Welcome to Ultimate Sleep Doctor. Tonight, we'll be exploring the quiet wonders within us. Deep inside every cell, a secret language unfolds, the story of DNA and genetics. It's a conversation written in the building blocks of life, a code that shapes who we are, tracing back through generations. Consider the intricate patterns, the elegant design woven into the very fabric of existence. There's a beautiful harmony to it all, a reflection of the universe itself. If you find this gentle exploration soothing, please consider subscribing and sharing this channel with anyone seeking a calm, curious mind. Just breathe. Let your thoughts drift like clouds across the evening sky. Let's begin. The air in the Cavendish Laboratory in Cambridge, England, in the early 1950s felt thick with anticipation, a quiet hum of brilliant minds wrestling with a puzzle that had stumped scientists for decades. The puzzle, of course, was DNA deoxyribonucleic acid, the very molecule that held the secrets of life's blueprints. Before 1953, it was known that chromosomes, the structures within our cells, carried genetic information. But the S-how remained stubbornly elusive. It wasn't a simple, linear code like a computer program. It had to be something far more intricate, something that could elegantly explain inheritance patterns, variations within species, and the astonishing diversity of life. The team working at the Cavendish, led by James Watson and Francis Crick, were making slow but steady progress, piecing together clues from a variety of sources. Watson, a young, energetic American, was a gifted experimentalist, driven by a relentless curiosity. Crick, a theoretical physicist with a profound understanding of molecular structures, brought a unique perspective, a framework for thinking about the molecule's potential form. Maurice Wilkins, a brilliant biophysicist, was also working at the lab, meticulously studying X-ray diffraction patterns of DNA fibers. These patterns, produced by bombarding DNA with X-rays, were like ghostly fingerprints, revealing information about the molecule's arrangement. However, the interpretation of these patterns was the crux of the challenge. The key to unlocking the DNA structure lay in the work of Rosalind Franklin and her graduate student, Raymond Gosling. Franklin, a quiet, meticulous chemist, had been tasked with producing high-resolution X-ray diffraction images of DNA. Using a specialized machine called a diffractometer, she and her team exposed DNA fibers to intense X-ray beams. The results were astonishing. Franklin's Codo 51, taken on April 25, 1952, was particularly striking. It showed a clear X-shaped pattern, indicating that DNA was not a uniform, fluffy material as previously assumed. Instead, it was a helical structure, spiraling around a central axis. This was a monumental discovery, providing the first concrete evidence for the molecule's three-dimensional shape. Initially, Watson, Crick, and Wilkins were largely unaware of the full significance of Photo 51. They were pursuing their own lines of inquiry, based on existing data about DNA's chemical composition, the sugar C-deoxyribose, the phosphate groups, and the nitrogenous bases adenine, guanine, cytosine, and thymine. Using this information, Crick began to construct a model of DNA based on the idea of a triple helix, a structure much like a spiral staircase. However, the data didn't quite fit. The X-ray patterns hinted at a more compact, double helix structure, where two strands of DNA wound around each other like a twisted ladder. Wilkins, recognizing the implications of Photo 51, shared a copy of the image with Watson and Crick without Franklin's knowledge or explicit permission. This was a contentious point, and the details surrounding the exchange remain somewhat debated to this day. While Watson and Crick acknowledged receiving the image, they felt that Wilkins hadn't fully appreciated its significance. It was the combined insights, the chemical properties gleaned by Watson and Crick, and the structural information revealed in Photo 51 that finally led them to the correct model. Working tirelessly, they realized that the X-shaped pattern indicated that the two strands of DNA were held together by hydrogen bonds, relatively weak bonds that could form between specific pairs of bases. Adenine always pairs with thymine, and guanine always pairs with cytosine. This base pairing rule was absolutely critical to the structure's stability. It meant that the two strands of the double helix ran in opposite directions, one running anti-clockwise, the other clockwise, a truly remarkable feat of molecular architecture. On February 28, 1953, Watson and Crick published their groundbreaking paper in the journal Nature, detailing their proposed structure of DNA. They elegantly explained how this double helix could accommodate the vast amount of genetic information required to direct the building of a living organism. The paper outlined the key features: the sugar-phosphate backbone providing structural support, the base pairing rules ensuring stability, and the helical shape maximizing the amount of information that could be stored within the molecule. The discovery of the double helix structure of DNA was one of the most important scientific breakthroughs of the 20th century. It instantly revolutionized our understanding of genetics, paving the way for countless advancements in medicine, agriculture, and biotechnology. The elegant simplicity of the double helix, a twisted ladder built from just 4 building blocks, continues to inspire awe and wonder, a testament to the power of scientific inquiry and the collaborative spirit of discovery. It wasn't just a structure, it was the key to unlocking the secrets of heredity and ultimately the very essence of life itself. The sheer volume of our body's trillions of cells, each teeming with activity, demands an incredible feat of engineering. Every cell needs a complete and identical copy of the instructions for building and operating itself. This isn't a haphazard process. It's a remarkably precise operation, and at the heart of it lies DNA replication. It's a story of teamwork, careful coordination, and a fundamental respect for the integrity of the genetic code. The cell begins with a complete set of DNA, neatly packaged into a chromosome. Before a cell divides, whether into 2 identical daughter cells during mitosis or into specialized cells during meiosis, this DNA needs to be duplicated. The process doesn't simply involve blindly shuffling the genetic information. It's a highly regulated and astonishingly accurate mechanism. The key players in this process are enzymes, the biological workhorses of the cell, and a carefully orchestrated sequence of steps. The first crucial step is unwinding the double helix. DNA, you remember, is structured like a twisted ladder, with 2 strands held together by chemical bonds. To copy it, this ladder must be separated. This is where an enzyme called helicase comes in. Helicase acts like a tiny zipper, breaking those hydrogen bonds that connect the 2 strands. It moves along the DNA, slowly unwinding it and creating a replication fork, a Y-shaped structure where the strands are now separated. Once the strands are separated, each serves as a template for building a new complementary strand. This is where the concept of base pairing becomes absolutely vital. Remember, adenine A always pairs with thymine ET, and cytosine C always pairs with guanine EG. It's like a matching game, but on a molecular level. An enzyme called DNA polymerase is the main builder of these new strands. However, DNA polymerase can't just start building randomly. It needs a starting point, an existing strand to latch onto. At the replication fork, there are special regions called origins of replication. These are specific sequences where the process begins. DNA polymerase begins adding nucleotides, the building blocks of DNA, to the new strand, following the base pairing rules. As it adds a new nucleotide, it removes the old one from the template strand, ensuring the sequence is maintained. This process is continuous, but not entirely smooth. Because DNA polymerase can only work in one direction, it can only add nucleotides to the 3 end of a growing strand. There are regions where the process is slightly less efficient. These areas, known as lagging strands, create short, discontinuous stretches of new DNA called Okazaki fragments. Imagine tiny, broken pieces being painstakingly assembled. Now, here's where another enzyme, DNA ligase, steps in. DNA ligase is the molecular glue. It acts like a tiny pair of scissors, cutting the weak links between the Okazaki fragments and then sealing them together to create a continuous strand. It's a critical step ensuring the stability of the replicated DNA. The lagging strand is synthesized in fragments, and the process is repeated continuously, with DNA polymerase and ligase working together tirelessly. The rate of replication is carefully regulated. It's not just a free-for-all. The cell ensures that the new strands are synthesized accurately, minimizing the risk of errors. Furthermore, the newly synthesized DNA strands aren't completely identical. One strand, called the leading strand, is synthesized continuously in the same direction as the replication fork moves. The other, the lagging strand, is synthesized in fragments, working backwards. This difference in synthesis direction contributes to the slightly different characteristics of the two strands, the parent and daughter strands, that eventually make their way into the new cells. The accuracy of DNA replication is remarkable. DNA polymerase has a built-in proofreading function. As it adds nucleotides, it checks to ensure that the base pairing is correct. If it detects an error, it can remove the incorrect nucleotide and replace it with the correct one. This happens so frequently that the error rate is incredibly low, far lower than would be expected from a purely random process. It's like having a meticulous editor constantly correcting mistakes. The entire process is tightly controlled by other enzymes and proteins, ensuring that replication occurs only when the cell needs it to. This control is essential for maintaining genetic stability and preventing mutations, changes in the DNA sequence that could lead to problems. The newly replicated DNA then distributes equally into the two daughter cells, ensuring that each receives a complete and accurate copy of the genetic instructions. This elegant mechanism, this remarkable ability of the cell to faithfully reproduce its DNA, is fundamental to life itself. It allows for growth, repair, and the passing of inherited traits, the very essence of heredity. It's a silent, tireless process, a remarkable feat of molecular engineering that underpins everything we know about biology. The precision of DNA replication is a constant reminder of the underlying order and beauty of the natural world. The blueprint of life, the intricate dance of heredity, doesn't unfold solely within the silent depths of the nucleus. While DNA holds the complete instructions, it's remarkably cautious about directly guiding the construction of proteins, the workhorses of the cell. This direct manipulation would be incredibly risky, prone to errors that could disrupt the delicate balance of cellular function. So, nature devised a clever intermediary, a temporary messenger, to carry the specific instructions needed for a protein's creation. This intermediary is RNA, and its creation from DNA is called transcription, a foundational process in gene expression. Transcription begins with a region of DNA that contains the gene we're interested in. Think of a gene as a specific chapter within a vast instruction manual. This chapter isn't meant for immediate action. Instead, it's carefully guarded. The first crucial step involves the unwinding of this DNA double helix. An enzyme, RNA polymerase, is like a tiny, highly specialized machine designed for this very task. It doesn't simply rip apart the DNA, it delicately separates the two strands, leaving a small portion of the DNA double helix intact, a carefully constructed bubble around the active site. RNA polymerase isn't just a simple copying device. It possesses a remarkable ability to recognize and bind to specific sequences on the DNA template strand. These sequences are called promoters, signaling the start of a gene. As it binds, the RNA polymerase begins to read the DNA sequence and, using this information, synthesizes a complementary RNA molecule. This RNA molecule is fundamentally different from DNA. DNA contains the bases adenine ZA, thymine T, guanine G, and cytosine C, while RNA uses uracil IUV in place of thymine. So, where DNA uses A and tRNA uses A and U, the process of building this RNA molecule is called elongation. RNA polymerase moves along the DNA template strand, adding RNA nucleotides one by one to the growing RNA chain. Each nucleotide added is carefully chosen based on the sequence of the DNA template. It's a precise step-by-step addition, driven by the inherent information encoded within the DNA. The newly synthesized RNA molecule is also complementary to the DNA template strand, meaning it mirrors the sequence of the DNA, but with the substitution of uracil for thymine. There are actually different types of RNA produced during transcription, each serving a distinct role. The RNA molecule being created in this initial stage is messenger RNA, or mRNA. mRNA is the direct copy of the gene's instructions. However, the process isn't simply a perfect duplication. A crucial modification happens during mRNA creation, the addition of a special cap at the beginning of the molecule and a tail at the end. These modifications protect the mRNA from degradation and aid in its transport from the nucleus to the ribosome, the site of protein synthesis. As the RNA polymerase continues its journey along the DNA template, it encounters sequences that signal the end of the gene. This signals the termination of transcription, and the RNA polymerase detaches from the DNA. The newly synthesized mRNA molecule, now carrying the genetic code for a specific protein, is released. Now, it's important to understand that the mRNA molecule is incredibly fragile. It's a temporary copy, a fleeting message. If it were to remain in the nucleus, it would be vulnerable to damage. That's why the mRNA quickly exits the nucleus and travels to the ribosomes, the protein-building factories of the cell. The mRNA molecule then delivers its message to the ribosome, where transfer RNAs to RNA molecules read the code and assemble the protein from amino acids. This process, translation, is the second crucial step in gene expression, following transcription. The beauty of this system is it's built in safeguards. If a mistake occurs during transcription, it's immediately corrected because the mRNA is a copy of the original DNA sequence. Furthermore, the entire process is tightly regulated, ensuring that only the necessary genes are expressed at the right time and in the right amount. This intricate control is essential for maintaining the health and function of an organism. Think of it like a carefully worded instruction manual being meticulously copied and delivered to the construction site. A slight error in the copy could lead to a flawed building. Transcription is a remarkably accurate and controlled process, showcasing the elegance of the biological world. It's the first step in a remarkable journey, a journey from the silent instructions within DNA to the active proteins that drive life itself. And it's a journey that continues, constantly shaping and defining who and what we are. The intricate dance of life, revealed by the secrets of DNA, isn't a solitary event. It's a collaborative performance, a carefully choreographed sequence involving several types of RNA. While DNA holds the master blueprint, RNA molecules act as the messengers, the workers, and the structural components that translate that blueprint into the proteins that build and operate every cell in our bodies. Let's delve into the three primary players: messenger RNA, transfer RNA, and ribosomal RNA, each with a distinct role and a unique contribution to this incredible process. Messenger RNA, or mRNA, is perhaps the most well-known type of RNA. Think of it as a temporary copy, a photocopy of a specific gene from the DNA master plan. This photocopying process, called transcription, which you'll recall was the first step in gene expression, occurs in the nucleus of the cell. The gene, A section of DNA is read, and a complementary RNA strand is created. This mRNA strand carries the genetic code from the nucleus to the ribosomes, the protein factories of the cell. The sequence of nucleotides in the mRNA is read in groups of 3. These are called codons, each codon specifying a particular amino acid. It's like a series of instructions, a recipe for building a protein. The more complex the protein, the longer and more intricate the mRNA sequence. Importantly, mRNA is relatively unstable. It's designed to be a transient messenger, quickly degraded after delivering its instructions, preventing the cell from being overwhelmed with unnecessary protein production. Different cells can produce different mRNAs, allowing them to synthesize different proteins, ultimately leading to the diversity of tissues and organs within a single organism. The accuracy of mRNA production is vital. Errors here can lead to the production of faulty proteins, potentially causing illness or disease. Now, let's turn our attention to transfer RNA, or tRNA. This molecule has a very specific and vital job. It acts as a delivery service, transporting individual amino acids to the ribosomes. Each tRNA molecule is designed to recognize a particular codon on the mRNA. Think of it as a key fitting into a specific lock. Each tRNA molecule has an anticodon, a sequence of 3 nucleotides that is complementary to a specific codon. When the ribosome encounters an mRNA codon, the appropriate tRNA molecule, carrying its attached amino acid, binds to that codon. This is where the actual building of the protein chain begins. There are numerous types of tRNA, each associated with a different amino acid and capable of recognizing a specific codon. The elegance of this system is remarkable. It ensures that the correct amino acids are added to the growing protein chain in the precise order dictated by the mRNA sequence. tRNA molecules are remarkably stable compared to mRNA, allowing them to participate repeatedly in the protein synthesis process. The efficiency of tRNA molecules is crucial for the rapid production of proteins within a cell. Finally, we have ribosomal RNA, or rRNA. This is the most abundant type of RNA in the cell, and it forms the structural and functional core of the ribosome itself. Ribosomes are complex molecular machines that read the mRNA sequence and facilitate the assembly of proteins. rRNA molecules, along with ribosomal proteins, make up the ribosome's 2 subunits, a large subunit and a small subunit. The small subunit binds to the mRNA, while the large subunit catalyzes the formation of peptide bonds between the amino acids. Think of the ribosome as a construction site and rRNA as the foundational framework and the machinery for assembling the final structure. RNA is crucial for the accurate translation of the mRNA code into a functional protein. It's not just a passive participant, it actively drives the chemical reactions necessary for protein synthesis. Furthermore, the amount of rRNA within a cell changes dramatically depending on the cell's activity level. More RNA is produced when protein synthesis is high. The structure of RNA is highly conserved across different species, reflecting the fundamental importance of this molecule in all living organisms. The interplay between these 3 RNA types, mRNA, tRNA, and rRNA, is a beautiful example of biological coordination. mRNA carries the blueprint, tRNA delivers the building blocks, and rRNA provides the machinery. This intricate system, known as translation, converts the information encoded in DNA into the proteins that perform nearly every function within our cells. It's a remarkably efficient and elegant process, and understanding the roles of these RNA molecules gives us a deeper appreciation for the complexity and wonder of life itself. The continuous cycle of transcription and translation, fueled by these RNA messengers, ensures that the instructions from our DNA are constantly realized, allowing us to grow, heal, and thrive. The constant activity within our cells, driven by these RNA molecules, is a testament to the astonishing precision and efficiency of the natural world. The blueprint of life, the DNA molecule, is often described as a code, and that's a remarkably accurate way to think about it. But what exactly is this code, and how does it work to build everything from a tiny bacteria to a towering redwood tree? It all boils down to a very specific relationship between the order of letters, the four chemical bases that make up DNA: adenine ZA, thymine ZT, cytosine C, and guanine ZG, and the sequence of amino acids that build proteins. Proteins, in turn, are the workhorses of the cell, carrying out almost every function imaginable. The magic begins with the genetic code itself, a set of instructions that translates these DNA sequences into a language of amino acids. This isn't a random alphabet, it's a carefully constructed system, a triplet code, meaning that 3 DNA bases always specify a single amino acid. Think of it like a 3-digit number. Any combination of 0, 1, and 2 will create a unique value. Similarly, 3 bases, such as CAT, TAG, or GCT, each represent a specific amino acid. There are actually 64 different possible combinations of 3 bases, and since there are only 20 standard amino acids, some triplets don't directly code for an amino acid at all. They act as start or stop signals, directing the protein building process. This code wasn't discovered overnight. Scientists, primarily through the work of Marshall Nirenberg and Heinrich Botstein in the early 1960s, painstakingly determined which DNA triplets corresponded to which amino acids. They used a clever experimental approach, feeding cells with different DNA sequences and analyzing the resulting proteins. By systematically varying the DNA and observing the changes, they cracked the code, demonstrating that the sequence of bases directly dictated the sequence of amino acids in the proteins. This was a truly revolutionary discovery, fundamentally changing our understanding of how genes control the characteristics of living organisms. Now, how does this translation actually happen? That's where transfer RNA, or tRNA, comes into the picture. tRNA molecules are tiny, specialized molecules, each uniquely shaped to bind to a specific amino acid. Each tRNA has a region that recognizes a particular triplet of DNA bases, its anticodon, and another region that attaches to a specific amino acid. These anticodons are perfectly complementary to the codons found in the messenger RNase, mRNA molecule, which is itself created from the DNA during transcription. The process begins with the mRNA molecule, created from a DNA template, attaching to a ribosome, a complex molecular machine responsible for protein synthesis. The ribosome moves along the mRNA, reading the codons one by one. As it encounters each codon, a tRNA molecule with the matching anticodon binds to it. The tRNA then delivers its attached amino acid to the ribosome. The ribosome links these amino acids together, forming a growing chain, one amino acid after another. This process is known as translation. As the ribosome continues to move along the mRNA, the chain of amino acids elongates, gradually forming the intricate 3-dimensional structure of the protein. The stop codons, which are DNA triplets that don't code for amino acids, signal the end of the message, and the newly formed protein is released. It's important to remember that a single gene, a segment of DNA that contains the instructions for making a specific protein, can be incredibly long. The genetic code ensures that only the necessary information is used to build the protein, and the process is remarkably precise. Errors can occur during transcription or translation, but cellular mechanisms often have quality control systems to correct them. The diversity of proteins generated from the same gene is astounding. The order of the DNA bases can be subtly altered, resulting in variations in the amino acid sequence. These small changes can have a significant impact on the protein's structure and function, leading to variations in the organism's traits. Everything from eye color to susceptibility to disease. The genetic code is the foundation upon which all biological diversity is built, a testament to the elegant and fundamental nature of life itself. It's a continuous, remarkable conversation between the information stored in DNA and the building blocks of life. A conversation that unfolds every moment within every living thing. The universe of genetics. Once you've grasped the basic dance of DNA, RNA, and proteins, reveals a fascinating and sometimes surprising element variation. It's not a static blueprint, it's a dynamic system, constantly susceptible to change. These changes, we call them mutations, are the raw material for evolution, and they're surprisingly common, occurring with every replication of DNA. They aren't necessarily bad. Many have no noticeable effect, but they introduce the possibility of altering the instructions encoded within our genetic code. Mutations arise in several ways. At their most fundamental, they begin with errors during DNA replication. DNA polymerase, the enzyme responsible for copying our genetic instructions, isn't perfect. It can occasionally misread a sequence or insert an incorrect nucleotide, a building block of DNA, into the new strand. The rate of these errors isn't constant. It's influenced by factors like the age of the cell and the availability of resources. Younger cells, undergoing rapid division, generally have a higher mutation rate than older, more stable cells. Think of it like carefully copying a long document. Even a diligent scribe might make a small mistake now and then, especially when rushing. Beyond simple replication errors, mutations can be triggered by external factors. Exposure to radiation, like ultraviolet, UV light from the sun, or X-rays, can damage DNA. These high-energy forms of radiation can directly break the chemical bonds holding the DNA strands together, creating single-stranded breaks, or even changing the shape of the bases themselves. Similarly, certain chemicals, known as mutagens, can interact with DNA and cause similar damage. Some mutagens are naturally occurring, like formaldehyde, while others are deliberately introduced in industrial processes or even found in some foods. Another category of mutations involves insertions and deletions. These are perhaps more dramatic changes. An insertion occurs when one or more extra nucleotides are added to the DNA sequence, while a deletion removes nucleotides. The impact of these changes depends entirely on where they occur. If a small number of bases are inserted or deleted, the reading frame of the gene may remain intact, and the resulting protein might be only slightly altered. However, if the insertion or deletion shifts the reading frame, that is, it causes the ribosome to read the codons incorrectly, it can result in a completely different, and likely non-functional, protein being produced. Imagine a sentence where a single word is misplaced, the entire meaning could be lost. Then, there are substitutions, the most frequent type of mutation. Here, one nucleotide is replaced by another. These substitutions can be categorized further. Transitions involve the replacement of a purine base, adenine or guanine, with another purine, or a pyrimidine base, cytosine or thymidine, with another pyrimidine. Transitions are often less disruptive than transversions, which involve the replacement of a purine with a pyrimidine or vice versa. The consequences of a mutation on the genetic information are hugely variable. Some mutations are silent. These occur in regions of DNA that don't code for protein, such as non-coding sequences. Because they don't influence the sequence of amino acids in a protein, they have no effect on the protein's function. Others might be synonymous, meaning they occur within a coding region but don't alter the amino acid sequence due to the redundancy of the genetic code. Several different codons can code for the same amino acid. However, many mutations are not silent. When a mutation occurs in a coding region and changes the sequence of amino acids, it can have significant effects on the protein's structure and function. A single amino acid change can alter the protein's folding, stability, or its ability to bind to other molecules. This, in turn, can disrupt the protein's role in a cellular process. For instance, A change in a protein involved in DNA repair could increase the risk of further mutations. The impact extends beyond individual proteins. Mutations can also alter the regulatory regions of genes, the areas that control when and where a gene is turned on or off. These changes can dramatically affect gene expression, leading to altered levels of protein production. It's important to remember that mutations aren't always detrimental. In some cases, they can provide a selective advantage. For example, a mutation that confers resistance to a disease or allows an organism to thrive in a new environment can be beneficial. Many of the genetic variations that contribute to human diversity, traits like eye color, hair type, and even susceptibility to certain diseases, are the result of accumulated mutations over generations. Furthermore, the study of mutations is fundamental to understanding evolution. The gradual accumulation of these changes over vast stretches of time is the driving force behind the diversity of life on Earth. Mutations are the source of new genetic information, providing the raw material upon which natural selection acts, favoring individuals with advantageous traits. Scientists use techniques like DNA sequencing to identify and analyze mutations, gaining insights into the genetic basis of diseases, the evolutionary history of species, and the very process of life itself. The ongoing conversation between DNA mutation and the environment shapes the future of every living thing. The intricate dance of DNA, as we've explored, isn't always a perfect, precise replication. Sometimes, the chromosomes, those beautiful, tightly wound strands of genetic information, experience disruptions, leading to changes in the number or structure of chromosomes. These irregularities, known as chromosomal aberrations, can have profound effects on an organism's development and health. We're going to delve into two particularly significant types, aneuploidy and polyploidy. Aneuploidy, a word that might seem a little daunting, simply means an abnormal number of chromosomes. It's the most common type of chromosomal aberration and often arises during the process of cell division, specifically meiosis, the division that creates sperm and egg cells. During meiosis, chromosomes normally pair up and then separate, ensuring that each new cell receives the correct number. However, this separation isn't always flawless. Non-disjunction, a fancy term for an error in chromosome segregation, can cause some cells to receive an extra copy of a chromosome, while others miss one entirely. Down syndrome is perhaps the most well-known example of aneuploidy. It's caused by trisomy 21, meaning an individual has 3 copies of chromosome 21 instead of the usual 2. This extra genetic material disrupts the normal developmental processes, leading to a range of characteristics, including intellectual disability, distinctive facial features, and increased susceptibility to certain health conditions. Similarly, Turner syndrome arises from monosomy X, a female individual possessing only one X chromosome instead of the usual 2. This condition can manifest with short stature, ovarian dysfunction, and other developmental differences. There are other examples of aneuploidy as well, Klinefelter syndrome, caused by an extra X chromosome in males, often associated with tall stature and reduced fertility, and trisomy 18, a more severe condition characterized by rapid growth in infancy followed by developmental delays and serious health complications. The severity of the effects depends heavily on which chromosome is involved and the quantity of the extra or missing genetic material. It's important to note that not all aneuploidies are immediately apparent. Some individuals with subtle chromosomal imbalances may remain undiagnosed until they experience health problems or have children with similar conditions. Diagnosis often relies on techniques like karyotyping, a laboratory procedure that analyzes an individual's chromosomes under a microscope. The accuracy of karyotyping has improved dramatically over time, enabling clinicians to identify a wider range of chromosomal abnormalities. Now, let's shift our focus to polyploidy, a considerably different type of chromosomal aberration. While aneuploidy involves a change in the number of individual chromosomes, polyploidy describes an organism that has more than two complete sets of chromosomes. Humans, thankfully, are naturally diploid, meaning we have two sets of chromosomes, one from our mother and one from our father. However, in several groups of organisms, polyploidy is the norm. Plants, in particular, exhibit a remarkable prevalence of polyploidy. Many important crop plants, like wheat, cotton, and potatoes, are tetraploid, possessing 4 sets of chromosomes. This wasn't an accident of evolution. It was intentionally cultivated through a process called hybridization, where 2 different species are crossed, and the resulting offspring is then treated with chemicals to encourage the development of more chromosome sets. The reasons for this manipulation are complex, but they primarily revolve around increased vigor, larger fruit sizes, and enhanced yields. A tetraploid plant, for instance, often produces larger seeds and a greater number of seeds per plant, leading to a significant boost in agricultural productivity. Beyond plants, polyploidy occurs in some animals, although it's far less common. Some species of fish and amphibians display varying degrees of polyploidy, often associated with adaptations to different environments. The evolutionary significance of polyploidy is fascinating. It's believed to have played a crucial role in the diversification of plant species, allowing for rapid adaptation to new ecological niches. It provides a vast pool of genetic variation, which natural selection can then act upon. Think of it as a genetic toolbox, offering a greater range of possibilities for generating new traits and characteristics. Interestingly, polyploidy in animals can sometimes lead to developmental abnormalities, highlighting the delicate balance required for proper development. The increased genetic material can overwhelm the developmental pathways, resulting in defects. However, in certain cases, polyploidy can be beneficial, contributing to larger body size and increased robustness. Understanding chromosomal aberrations, particularly aneuploidy and polyploidy, reveals a fascinating aspect of the genetic landscape. These irregularities, though often associated with challenges, also demonstrate the remarkable plasticity of the genome and the diverse ways in which life can adapt and evolve. The precise choreography of chromosome division, a fundamental process of life, can sometimes stray from its intended path, yet the consequences, from the distinctive features of Down syndrome to the bountiful harvests of polyploid crops, contribute significantly to the incredible variety of life on Earth. The story of DNA is, at its heart, a tale of instruction. We've explored how that long, twisting ladder, this remarkable molecule, holds the blueprints for building every living thing. From the smallest bacterium to the tallest redwood, we've seen how variations in those blueprints, or mutations, can alter those blueprints, sometimes leading to changes in an organism. And we've acknowledged that sometimes errors occur during cell division, creating cells with a different number of chromosomes. But there's a layer of control, a subtle but incredibly powerful influence operating errors within the DNA itself, a way to turn genes on and off, to modify their activity without changing the underlying sequence. This is the realm of epigenetic modifications, and it's a story of communication, not just of instruction. Think of DNA as a library. The sequence of letters A, T, C, and G represents the books on the shelves. But those books aren't just passively sitting there. They can be marked, highlighted, or even rearranged on a bookmark. These aren't changes to the text itself, but they drastically alter how the information is read and utilized. Epigenetic modifications are like those bookmarks, signals that tell cells which parts of the DNA should be accessible and which should remain hidden. The primary mechanisms of these signals are chemical modifications to DNA and the proteins that wrap around it. DNA itself can be chemically altered through processes like DNA methylation. Imagine tiny little tags, methyl groups, being added to specific spots on the DNA strand. In many cases, when these tags are added, they silence the gene located nearby. It's like putting a "Do Not Read" sticker on a book. The more methyl groups attached, the stronger the silencing effect. This isn't a permanent change to the DNA sequence. It's a change in how the cell reads the sequence. But DNA methylation isn't the only story. Histones, the proteins that package and organize DNA into tightly wound structures called chromatin, also play a crucial role. DNA doesn't just float freely in the cell. It needs to be neatly arranged to be accessible. Histones can be modified by adding chemical tags like acetyl groups. These modifications are the opposite effect of methylation. They generally loosen the chromatin structure, making the DNA more accessible to the machinery that reads genes. Think of it as gently unfolding a book, making it easier to access the information inside. These histone modifications aren't isolated events. They occur in complex patterns, often acting as a kind of cellular language. Different combinations of modifications can create distinct states of chromatin, influencing which genes are active or inactive. It's like having a complex code, where a particular sequence of modifications communicates a specific message to the cell. Perhaps the most fascinating aspect of epigenetic regulation is its ability to be passed down through generations. Scientists have discovered that some epigenetic marks, particularly DNA methylation patterns, can be inherited. This means that experiences a parent has perhaps a diet rich in certain nutrients or exposure to environmental stressors could, in a way, influence the development of their offspring, even without any changes to the DNA sequence itself. This doesn't mean that children are clones of their parents, but their development can be subtly affected by these inherited epigenetic marks. Research has shown links between maternal diet during pregnancy and the epigenetic profiles of the child, influencing traits like metabolism and even susceptibility to certain diseases. For example, studies on rats demonstrated that restricting a mother's diet during pregnancy could lead to reduced metabolic activity in the offspring, even if the pups were later fed a normal diet. The epigenetic changes established during that period seem to prime the offspring for a similar lifestyle. The field of epigenetics is still relatively young, and scientists are constantly uncovering new details about these intricate regulatory mechanisms. It's becoming increasingly clear that epigenetic modifications aren't just about turning genes on and off. They're involved in a wide range of processes, including development, aging, and disease. Abnormalities in epigenetic regulation have been linked to several disorders, including cancer, autoimmune diseases, and neurological conditions. Interestingly, epigenetic modifications are dynamic. They can change in response to environmental cues. This means that our experiences can actually shape our biology, and that our cells retain a memory of past events. This also opens up exciting possibilities for therapeutic interventions. Imagine being able to reset abnormal epigenetic patterns that contribute to disease, effectively reprogramming cells back to a healthier state. The intricate dance of DNA, histones, and epigenetic marks represents a profoundly sophisticated system of control. It's a reminder that life isn't just about the sequence of information, but also about how that information is interpreted and utilized. It's a testament to the amazing adaptability and resilience of living organisms, a story of control beyond the letters themselves, a story that continues to unfold with every new discovery in the fascinating world of epigenetics. The story of DNA isn't just about the grand, famous genes, the ones that code for building proteins, the blueprints for everything from eye color to enzyme function. It's a much more intricate and surprisingly crowded place. For a long time, Scientists largely dismissed the vast stretches of DNA that don't seem to do anything directly. They called them junk DNA. And for a while, it felt like a shameful secret hidden within our genetic code. But recent discoveries have dramatically shifted our understanding, revealing that this non-coding DNA plays a remarkably vital role in orchestrating the symphony of life within every cell. Within these seemingly empty spaces lie fascinating structures, particularly introns and exons, which are central to how genes are actually used. Think of a gene like a carefully written paragraph in a complex book. The exons are the key sentences, the parts that ultimately get copied and used to create a specific protein. However, this paragraph isn't written in a continuous block. It's interrupted by sections called introns. These are like temporary, placeholder sentences or notes that don't contribute to the final message. The process of making a protein from a gene is called transcription, and it doesn't simply copy the entire gene. Instead, RNA polymerase, the enzyme responsible for copying the DNA, binds to the gene and begins transcribing only the exons. These exon sequences are then spliced together, much like editing a document, removing the introns, to create a continuous strand of messenger RNA, mRNA. This mRNA molecule then carries the instructions out of the nucleus and into the ribosomes, where the protein is finally built. This splicing process is incredibly precise, ensuring that only the correct exon sequences are combined to create the desired protein. Different combinations of exons can even lead to different proteins being produced from the same gene, a phenomenon known as alternative splicing, dramatically expanding the potential diversity of the proteome, the total set of proteins expressed by an organism. But the story doesn't end with introns and exons. The expanding role of regulatory sequences reveals that the non-coding DNA isn't just about gene structure. It's about controlling Fv when and Fv where genes are expressed. These regulatory sequences, scattered throughout the genome, act like switches and dials, fine-tuning gene activity. Promoters, for instance, are located just upstream of a gene and signal to the cell where and when the gene should be transcribed. Enhancers, which can be located far away from the gene they regulate, boost the activity of promoters, amplifying the expression of a gene in response to specific signals. These sequences don't code for proteins themselves, but they profoundly influence the cellular machinery, directing the flow of information with exquisite precision. The discovery of long non-coding RNAs, lincRNAs, has further complicated and enriched our understanding. These RNA molecules, much longer than typical messenger RNAs, don't directly code for proteins, yet they play a multitude of roles. Some lincRNAs act as guides, directing proteins to specific locations within the genome, like tiny GPS systems. Others act as scaffolds, bringing together different proteins to form complexes that regulate gene expression. Still others can silence genes entirely, preventing them from being turned on. The organization of non-coding DNA isn't random. It's often arranged in elaborate structures called topologically associating domains, or TADs. Imagine these TADs as self-contained communities within the genome, where DNA sequences tend to interact with each other, but rarely with sequences outside of the domain. This organization helps to compartmentalize gene regulation, preventing interference between genes that are distant on the chromosome. Furthermore, research has demonstrated that the sequence of non-coding DNA itself can influence gene expression. Variations in these sequences, sometimes referred to as epigenetic marks, can affect how tightly or loosely a gene is packaged, thereby impacting its accessibility to the machinery involved in transcription. These epigenetic marks, while not altering the DNA sequence itself, can be inherited across generations, contributing to the inheritance of traits and characteristics. The study of non-coding DNA is a rapidly evolving field, and scientists are continually uncovering new functions and complexities. It's becoming increasingly clear that the vast majority of our genome isn't junk at all. Instead, it's a dynamic and incredibly sophisticated regulatory network, constantly adapting and responding to the needs of the cell. The intricate interplay between coding and non-coding DNA represents a level of complexity that was largely unforeseen and continues to reshape our understanding of what it means to be human, a testament to the astonishing ingenuity of the biological world. As we delve deeper into the mysteries hidden within this non-coding landscape, we unlock a new appreciation for the elegance and control underlying the very essence of life itself. The late 20th century held a quiet, determined ambition, a truly grand undertaking that would reshape our understanding of life itself. It began with a simple question: what exactly is the instruction manual for being human? For centuries, scientists had known that genes carried hereditary information, but the complete sequence, the entire blueprint, remained a tantalizing, almost mythical goal. This was the impetus behind the Human Genome Project, an international collaborative effort that began in 1990 and ultimately aimed to produce a complete, accurate map of every single nucleotide, every A, T, C, and G that makes up the human DNA. The initial goals of the project were multifaceted. Primarily, it sought to determine the precise order of the 3 billion base pairs that constitute the human genome. Beyond simply sequencing, the project aimed to identify all of the genes within that sequence, an estimated 20,000 to 25,000 at the time. Researchers also hoped to discover how these genes work together, understanding the complex networks that governed our development, health, and susceptibility to disease. Crucially, the project wasn't just about academic curiosity. It was anticipated to have profound implications for medicine. Understanding the genetic basis of diseases would pave the way for more targeted therapies, personalized medicine, and improved diagnostics. However, achieving this monumental goal proved to be extraordinarily challenging. The human genome is, quite simply, vast. Sequencing such a large and complex molecule presented immense technical hurdles. Early sequencing methods, primarily based on chain termination chemistry, were slow, unreliable, and prone to errors. Imagine trying to read a massive library filled with millions of books, but each book was printed with numerous typos and gaps. This was the reality of early sequencing, requiring scientists to painstakingly piece together fragments of DNA, often millions of times before a complete picture could emerge. Furthermore, the DNA itself was remarkably complex. It wasn't just a straight line of letters. It was intricately folded and packaged into chromosomes. The way this DNA was organized, its three-dimensional structure, played a critical role in gene regulation, a concept explored in earlier parts of this narrative. Understanding how this structure influenced gene expression was a major hurdle. The project needed to develop sophisticated techniques to not only read the sequence, but also to characterize the physical arrangement of the genome within cells. The initial approach involved breaking the human genome into smaller, manageable pieces, known as contigs, and then assembling them using overlapping sequence reads. This process, often likened to solving a giant jigsaw puzzle, relied heavily on computational power and sophisticated algorithms. Teams of scientists, including biologists, computer scientists, and mathematicians, worked tirelessly to analyze the data, identify matches, and construct increasingly accurate assemblies. There were significant delays and setbacks, including a major contamination event in 1999 that forced the deletion of a large portion of the sequence, a stark reminder of the delicate and exacting nature of the work. Despite these challenges, the project made remarkable progress. In 2000, the International Genome Sequencing Consortium announced the completion of the human genome sequence. This wasn't a perfect map, it was a draft sequence, representing the best possible reconstruction based on the available data. However, it was a monumental achievement. The draft sequence was released publicly, making it accessible to researchers worldwide. The initial findings from the draft sequence were immediately transformative. It confirmed many of our existing assumptions about gene numbers and genome organization. It revealed the surprising extent of non-coding DNA, the vast stretches of sequence that don't directly translate into proteins, and highlighted its crucial role in regulating gene expression. Scientists discovered previously unknown genetic variations, including single nucleotide polymorphisms, SNPs, which are variations in a single DNA base, and are now recognized as major contributors to individual differences in traits and disease susceptibility. The project also identified many new genes involved in fundamental biological processes, such as development, immune response, and aging. These discoveries provided a foundation for further research and fueled the burgeoning field of genomics. It established a benchmark for future genome sequencing projects, demonstrating the feasibility of creating a complete human genome sequence within a relatively short time frame. The completion of the Human Genome Project marked a turning point in biology. It ushered in an era of unprecedented access to genetic information, transforming research and offering the potential to unlock the secrets of human health and disease. The draft sequence served as a springboard, leading to increasingly refined and complete assemblies and continuing to fuel ongoing research and innovation today. The legacy of the Human Genome Project isn't just a sequence of letters. It's a story of collaboration, perseverance, and a profound shift in our understanding of what it means to be human. The secrets held within our DNA aren't just about individual genes. They're interwoven in a complex tapestry. It's like a vast, intricate puzzle where pieces, genes, often sit close together on a chromosome. And sometimes, these nearby genes tend to be inherited together. That's where the idea of genetic linkage mapping comes in, a way of figuring out how genes are arranged and how they influence each other's inheritance. It's a technique that relies heavily on the oldest, most persistent form of family history, the family tree. The principle behind linkage mapping began with careful observations made by Thomas Hunt Morgan and his colleagues at Columbia University in the early 20th century. They were studying fruit flies, Drosophila melanogaster, because they bred quickly and their genetics were relatively simple compared to humans. Morgan and his team noticed a puzzling trend. Genes that were located close together on a fruit fly chromosome frequently appeared together in families. If a fly carried a gene for white eyes, it was almost always accompanied by a gene for curled wings. And these traits weren't inherited independently like Mendel's peas had suggested. Instead, they were passed down as a unit. This observation led to the understanding that genes don't separate randomly during meiosis, the process that creates sex cells. The physical proximity of genes on the chromosome prevents them from separating. The closer two genes are located, the less likely they are to be inherited independently. It's a fundamental concept. The closer the genes are, the stronger the linkage. To build a linkage map, researchers painstakingly constructed family trees, tracing the inheritance of specific traits through multiple generations. They'd start with a family displaying a clear, consistent pattern of inheritance for a particular characteristic, perhaps a disease or a physical feature. For example, a family might consistently show that a male with a certain eye color also had a higher incidence of a specific heart condition. By tracking these traits across several generations, they could build a strong case for a genetic link. The more families with similar inheritance patterns they could gather data from, the more accurate their linkage map would become. The strength of the linkage was quantified using a concept called recombination frequency. Recombination frequency is the proportion of offspring that show a new combination of traits, a recombination event, compared to the number of offspring that inherit the traits exactly as their parents did. A low recombination frequency indicates a strong linkage between two genes. Conversely, a high recombination frequency suggests the genes are far apart on the chromosome and therefore inherited independently with greater frequency. The process involved carefully recording the inheritance of various traits, some simple, like coat color in dogs, and others more complex, such as height or blood type. The key was to identify traits where inheritance patterns were reliably consistent within families. Once enough families were studied, the researchers could estimate the recombination frequency between different genes. These recombination frequencies were then used to create a map, a linear arrangement of genes along a chromosome, with the genes closest together having the lowest recombination frequency. Imagine a road map. Cities close together are connected by short, direct routes, while cities farther apart have longer, more winding roads. Similarly, genes close together on a chromosome tend to be inherited together because the road between them is shorter. However, the road isn't perfectly straight. It's punctuated by regions of higher recombination frequency, areas where the chromosome breaks and rejoins, creating new combinations of genes. The technique wasn't limited to fruit flies. As genetic technology advanced, researchers began applying linkage mapping to humans and other organisms. By studying families with inherited diseases, they could pinpoint the specific chromosomal location of the disease-causing gene. For example, early linkage mapping studies in families with cystic fibrosis helped identify the location of the faulty gene on chromosome 7. This information is incredibly valuable because it allows for more accurate genetic testing, carrier screening, and ultimately, the development of targeted therapies. Furthermore, linkage mapping isn't just about identifying single genes. It can also be used to identify clusters of genes that are inherited together, suggesting they may be involved in the same biological pathway or process. By examining the patterns of inheritance of multiple traits simultaneously, researchers can build a more detailed picture of the genetic architecture of a particular organism. It's a foundational technique that continues to evolve with the development of new technologies, such as whole genome sequencing, providing a deeper understanding of the intricate relationships between genes and inheritance. The family tree, in its simplest form, remains a crucial tool in unlocking these secrets. The blueprint of life, the DNA that carries our instructions, isn't a perfectly uniform document. It's more like a vast, sprawling library filled with countless editions of the same book, the human genome. And within these different editions, there are tiny variations, differences that exist between individuals. These variations, while subtle, hold a remarkable amount of information about our ancestry, our health, and our susceptibility to a huge range of conditions. The most common of these variations are called single nucleotide polymorphisms, or SNPs. A SNP, simply put, is a change in a single building block of DNA. DNA is made up of 4 chemical bases: adenine A, thymine T, cytosine C, and guanine G. SNPs occur when a single base pair, meaning 2 adjacent bases, differs between individuals. It's like finding a single misplaced comma in a gigantic book. It doesn't fundamentally change the meaning of the whole text, but it can have subtle consequences, especially when multiplied across the genome. Scientists estimate that the average human genome contains around 20 to 30 million SNPs, and most people have at least one SNP that differs from their siblings or parents. The beauty of SNPs lies in their incredible abundance. Because they are so common, they've become the primary tool researchers use to study the intricacies of genetic variation. Instead of trying to analyze every possible difference across the genome, which would be an impossibly complex task, Scientists focus on SNPs because they are consistently available and relatively easy to measure. Think of it like this. If you were trying to study the color of a vast field of wildflowers, it would be far simpler to count the number of red, yellow, and blue flowers than to meticulously examine the pigment composition of every single petal. The way SNPs are identified is straightforward. Researchers use techniques like DNA sequencing, which essentially reads the order of the bases in a DNA sample. When comparing the DNA of different people, they can pinpoint the SNPs that are present in one individual but absent in another. These differences become a unique genetic fingerprint, a sort of barcode that can be used to distinguish between people. The applications of SNPs are truly astonishing and are already transforming numerous fields of study. One of the most impactful areas is in medical research. Scientists are using SNPs to investigate the genetic basis of diseases. Many diseases, like heart disease, diabetes, and certain cancers, have a genetic component, meaning that variations in genes can increase a person's risk of developing the condition. By identifying the SNPs associated with these diseases, researchers can develop personalized medicine approaches. This doesn't necessarily mean predicting a person's fate with absolute certainty, but rather understanding their genetic predisposition and tailoring treatment strategies accordingly. For example, a person with a specific SNP linked to increased risk of developing a certain type of cancer might be advised to undergo more frequent screenings or be prescribed preventative medications. Furthermore, SNPs are being used to study ancestry and population history. By analyzing the SNPs present in DNA samples from people around the world, scientists can trace human migration patterns and understand how different populations have diverged over time. It's like piecing together a complex family tree, but instead of tracing lineages through generations, they're tracing them through variations in DNA. The geographic distribution of certain SNPs can provide clues about the routes our ancestors took and the interactions they had with other populations. Another exciting area of research is in agriculture. SNPs are being used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. By identifying SNPs that contribute to desirable traits, breeders can select and cross plants with those traits, accelerating the development of improved varieties. This has the potential to increase food production and ensure food security for a growing global population. The study of SNPs isn't without its challenges. Interpreting the function of a SNP, understanding how it affects the expression of a gene or interacts with the environment, can be complex. Many SNPs have no known effect, while others might have subtle influences that are difficult to detect. As technology advances, researchers are developing more sophisticated methods to analyze SNPs and understand their roles in health and disease. Beyond these established applications, the field of SNP research is constantly evolving. Scientists are exploring the potential of SNPs to diagnose diseases earlier, predict treatment responses, and even develop new therapies. The ongoing research into SNPs represents a powerful approach to unraveling the mysteries of life and improving human health. It's a story written not in a single, flawless book, but in the countless, subtly varied editions of the human genome, a testament to the beautiful complexity of our genetic heritage. The world of genetics has always held a certain magic, a hidden language written in the very building blocks of life. For decades, scientists dreamed of directly manipulating that language, of correcting errors, or adding new instructions to the blueprints of living things. That dream took a monumental leap forward with a system borrowed from a remarkable natural defense mechanism found in bacteria, a system now known as CRISPR-Cas9. At its heart, CRISPR-Cas9 isn't a complicated machine, but a surprisingly elegant and precise tool. It all begins with Cas9, an enzyme, a tiny protein that acts like a pair of molecular scissors. But Cas9 doesn't just cut randomly. It's guided by a short piece of RNA, often called a guide RNA, which is designed to match a specific sequence of DNA within a gene. Think of the guide RNA as a tiny, incredibly accurate GPS, directing the Cas9 enzyme to the exact location it needs to target. The process starts with the creation of this guide RNA. Scientists design it carefully, selecting a sequence that corresponds to the gene they want to edit. This guide RNA then forms a complex with the Cas9 enzyme. When the complex finds its matching DNA sequence, Cas9 makes a precise cut, a clean break, in the double helix. Once the DNA is cut, the cell's natural repair mechanisms kick in. There are two primary ways the cell can respond. Sometimes, the cell simply repairs the break, but this repair process can introduce small insertions or deletions of DNA, effectively disrupting the gene's function. Other times, the cell uses a supplied piece of DNA, a template, as a guide to rebuild the cut sequence. This allows scientists to insert a completely new DNA sequence, replacing the original or adding entirely new genetic information. The beauty of CRISPR-Cas9 lies in its adaptability and relative simplicity. Before this technology, editing genes was a laborious and often imprecise process. CRISPR-Cas9 dramatically reduced the time and effort required, opening up a vast range of possibilities. Researchers are now using it to tackle diseases in a way that was previously unthinkable. One of the most exciting applications is in treating genetic diseases. For conditions like cystic fibrosis, sickle cell anemia, and Huntington's disease, which are caused by mutations in single genes, CRISPR-Cas9 offers the potential to correct those mutations directly. Clinical trials are underway exploring the use of CRISPR-Cas9 to treat these diseases, with early results showing promising signs. Scientists are working on delivering the CRISPR-Cas9 system to specific cells in the body, targeting affected tissues and minimizing potential side effects. Beyond human health, CRISPR-Cas9 is revolutionizing agriculture. Scientists are using it to enhance crop yields, improve nutritional content, and increase resistance to pests and diseases. Imagine developing rice that is naturally fortified with vitamin A, or wheat that is resistant to drought conditions, all achievable through targeted gene editing. Similarly, researchers are exploring the use of CRISPR-Cas9 to create livestock with improved traits, such as increased meat production or enhanced disease resistance. However, the power of CRISPR-Cas9 also raises significant ethical considerations. The ability to edit genes isn't without potential risks and unintended consequences. Because the system targets DNA sequences, there's a possibility of off-target effects where the Cas9 enzyme cuts at locations in the genome that are similar, but not identical, to the intended target. This could lead to unintended mutations with unpredictable outcomes. Researchers are continuously working to improve the specificity of the system and develop safeguards to minimize off-target effects. Another major concern revolves around the potential for germline editing, making changes to genes that can be passed down to future generations, while theoretically offering a permanent solution to inherited diseases. Germline editing raises profound ethical questions. It essentially alters the human gene pool, with long-term consequences that are difficult to fully predict. There's a broad consensus that germline editing should be approached with extreme caution, and in many jurisdictions, it is currently prohibited for human applications. The discussion around CRISPR-Cas9 isn't just about science, it's about values and society. Questions arise about who has access to this technology, how it should be regulated, and what constitutes a desirable trait. The potential for designer babies, individuals selected and genetically modified for specific characteristics, is a recurring theme in the debate. Robust public discourse involving scientists, ethicists, policymakers, and the public is crucial to navigating these complex issues and ensuring that this powerful technology is used responsibly and for the benefit of all. The ongoing evolution of Cas9 is a testament to the dynamism of scientific discovery, but it's equally a reminder of our responsibility to wield such power with wisdom and foresight. As researchers continue to refine the technology, the conversation surrounding its use will undoubtedly evolve alongside it, shaping the future of genetics and its impact on our world. The story of DNA is a grand one, woven through the cells of every living thing. We've been tracing the connections between genes, exploring how they're passed down through families, and even touching upon the incredible possibilities of altering them. But there's another, incredibly ancient, thread within our genetic tapestry, a thread carried solely by the women in our family line, and it's a story that stretches back millions of years. This thread is made of mitochondrial DNA, or mtDNA for short, and it holds secrets about our deepest ancestry. Unlike the DNA found in the nucleus of our cells, the DNA that determines our eye color, hair texture, and susceptibility to certain diseases, mtDNA, lives in the mitochondria, tiny bean-shaped structures within nearly every cell. These mitochondria are the powerhouses of the cell, diligently converting the energy from our food into a usable form. Remarkably, each mitochondrion contains its own small, circular chromosome, a miniature genome completely separate from the main DNA blueprint. This unique origin means mtDNA doesn't come directly from our parents. Instead, it was inherited from our maternal grandmother and her grandmother before her, all the way back to the very first female ancestor of our lineage. The inheritance of mtDNA is a fascinating example of maternal inheritance. When a sperm fertilizes an egg, the sperm contributes its nuclear DNA. However, the egg provides the sole source of mitochondria and therefore the mtDNA. As a result, every child of a woman receives only her mtDNA, creating a direct, unbroken line of inheritance tracing solely through the female side of the family. This is why a DNA test focusing solely on mtDNA can tell you a great deal about your maternal lineage, even if it doesn't reveal information about your father's family. Now, let's consider the nature of mtDNA itself. Because it's a circular DNA molecule, it replicates somewhat differently than nuclear DNA. This difference makes it more prone to accumulating mutations over vast stretches of time. Nuclear DNA, with its double-stranded structure and intricate repair mechanisms, is considerably more stable. Consequently, mtDNA carries a much higher mutation rate. These mutations or changes in the DNA sequence are incredibly valuable to scientists studying human evolution. Think of it like a very old, well-worn road. The more traveled it is, the more potholes and worn patches you'll find. Similarly, mtDNA, due to its ancient origins and higher mutation rate, has accumulated a vast collection of variations across different individuals. Scientists can analyze these variations, we call them polymorphisms, and use them to build a detailed family tree extending back thousands of years. Some polymorphisms are very common, while others are incredibly rare, providing a unique fingerprint for each individual and their maternal lineage. The study of mtDNA mutations isn't just about tracing family trees. It has profound implications for understanding human migration patterns and the origins of diseases. Certain mutations are associated with specific geographic populations, suggesting that groups of people migrated from particular areas and carried those mutations with them. For example, a particular mutation found predominantly in populations across Southeast Asia has been linked to the spread of rice cultivation. The mutation likely originated in a single location and spread as people migrated with the crop. Furthermore, some mtDNA mutations increase the risk of certain diseases. Mutations affecting the enzymes involved in mitochondrial energy production can lead to mitochondrial diseases, often characterized by muscle weakness, neurological problems, and heart dysfunction. These diseases typically manifest when the mutation is present in both copies of the mtDNA, which is rare. However, having just one copy of a mutated mtDNA can also lead to health issues, even if they aren't immediately apparent. Scientists are actively researching mtDNA mutations linked to conditions like diabetes, Parkinson's disease, and even autism, exploring how these changes might affect cellular function. The study of mtDNA has even helped solve some historical mysteries. By analyzing mtDNA from ancient human remains from Hutzai the Iceman, who lived over 5,000 years ago, to the remains of early farmers in Europe, scientists can determine the geographic origins of these individuals and their relationships to modern populations. It's like having a genetic witness to past events, offering a more direct connection to our ancestors than traditional historical records often provide. The genetic information preserved in mtDNA provides a window into the deep history of humanity, revealing patterns of migration, adaptation, and survival that have shaped our species. As we continue to explore the secrets held within mtDNA, we uncover new insights into the intricate dance of inheritance and the remarkable journey of our ancestors. This tiny, ancient thread, passed down through the women in our family, offers a powerful testament to the enduring legacy of our maternal lineage, a legacy that stretches back through time and connects us to the very beginnings of our human story. The blueprint of life, DNA, isn't always a straightforward story. Sometimes it tells a tale with a little extra twist, a hidden player that dramatically alters the inheritance pattern. This twist often involves the X and Y chromosomes, the sex chromosomes, and the fascinating concept of X-linked inheritance. It's a difference in how traits are passed down from parent to child, and it's particularly noticeable when considering conditions affecting males. The key to understanding X-linked inheritance lies in recognizing that females typically have two X chromosomes, XX, while males have only one XY. This difference is critical. Let's consider a gene located on the X chromosome. Because males only possess one X, they don't have a second copy to mask a potentially harmful mutation. If a male inherits a faulty version of this gene, the effect is almost always visible. It's like having only one key to a lock. If that key is broken, the door won't open. Now, let's look at how this plays out with recessive X-linked traits. Many diseases, such as hemophilia and red-green color blindness, follow this pattern. These conditions are caused by mutations in genes located on the X chromosome. Crucially, the faulty gene needs two copies, one from each parent, to fully express itself. Because males only have one X, they're much more likely to be affected. A female with her two X chromosomes might be a carrier. A carrier has one normal X chromosome and one X chromosome carrying the mutated gene. She doesn't herself exhibit the disease symptoms because the normal X chromosome compensates for the faulty one. However, she has a 50% chance of passing the mutated X chromosome onto her children, regardless of their sex. Let's illustrate this with a fictional example. Let's call the gene color sense. A male with color sense affected color sense F inherits the faulty version from his mother. Since he only has one X chromosome, he will definitely show the symptoms of color blindness. A female can be either unaffected of color sense NEF, a carrier of color sense CMNI, or affected color sense F. If a carrier female has a child with a male who is unaffected, there's a 50% chance the child will be unaffected as inheriting the normal X chromosome, a 25% chance the child will be a carrier's, inheriting one normal and one affected X chromosome, and a 25% chance the child will be affected as inheriting two affected X chromosomes. The patterns of inheritance become even more interesting when considering dominant X-linked traits, though they are less commonly observed in diseases. A dominant X-linked trait only needs one copy of the mutated gene to be expressed. Again, this is more prevalent in males. If a male inherits a single copy of a dominant X-linked gene, he will definitely exhibit the associated trait. A female carrier, having one normal and one affected X chromosome, will also express the trait. Consider another hypothetical gene, strong-build. If a male inherits strong-build, F, he will exhibit the characteristics associated with the condition, such as unusually tall stature. A female carrier will also display those same characteristics because she has two copies of the affected gene. This is due to her two X chromosomes carrying the faulty version. The probability calculations in X-linked inheritance can seem complex, but they reflect the fundamental difference in chromosomal makeup between males and females. The ratio of males to females affected by X-linked recessive conditions is typically around 4 males affected for every 1 female affected. This isn't a coincidence, it's a direct consequence of the chromosomal disparity. Furthermore, X inactivation plays a significant role in determining the specific phenotype observed in females. After fertilization, one of the two X chromosomes in female cells is randomly inactivated. This process, called X inactivation, ensures that females don't have twice the amount of X-linked gene products, which could be detrimental. The inactivation is largely random, but it tends to be more prevalent in the mother's cells than in the father's. This random inactivation further contributes to the variability in expression of X-linked traits in females. Some cells may silence the faulty gene, while others retain it, leading to a range of symptoms or a complete absence of symptoms in carriers. Understanding X-linked inheritance is a cornerstone of genetic counseling and predicting the inheritance of conditions such as Duchenne muscular dystrophy and some forms of osteoporosis. It highlights how the simple sequence of DNA can be profoundly impacted by the intricacies of sex chromosome inheritance. The story of DNA isn't just about the code itself, but also about how that code is interpreted and transmitted, shaped by the very structure of the chromosomes. The subtle shifts in inheritance patterns, like those found in X-linked traits, demonstrate the remarkable complexity and elegance of the biological world. The world of genetics is fascinating, isn't it? We've spent a good while exploring how our blueprints, the instructions for building us, are passed down through generations. But sometimes, these blueprints contain surprises variations that can influence our health, our risk of certain diseases, and even the way we look. This is where genetic counseling comes in, acting like a gentle guide through the complex landscape of our inherited information. Genetic counselors are trained professionals, typically nurses or other healthcare workers with specialized knowledge in genetics, psychology, and counseling. They're not scientists who perform DNA tests themselves. Instead, their role is to help individuals and families understand the implications of genetic information, whether it's from a test result or a family history. It's about translating the complex language of genes into something that's meaningful and manageable. The principles guiding genetic counseling are built around respect for autonomy, confidentiality, and non-judgment. Everyone has the right to make their own decisions about genetic testing and its implications. Genetic counselors facilitate this process by providing information, exploring concerns, and helping individuals weigh the potential benefits and risks. Confidentiality is paramount. The information shared during counseling remains private and protected. Finally, counselors approach each situation with empathy and understanding, recognizing that genetic information can be profoundly personal and emotionally charged. The scope of genetic counseling is incredibly broad, touching upon many different areas. Initially, much of the work focused on single gene disorders, conditions caused by a mutation in a single gene. Cystic fibrosis, sickle cell anemia, Huntington's disease, these were, and still are, major areas of counseling. Genetic counselors would help families understand the inheritance pattern of these diseases, often following Mendel's laws, although sometimes with more intricate patterns. Assess the risk of future generations inheriting the condition, and discuss treatment options. However, the field has expanded dramatically with the advancements in genetic testing. Today, genetic counseling is routinely offered to individuals and families who've undergone carrier screening. Carrier screening involves testing to determine if someone carries a gene variant that could be passed on to their children, even if they don't have the disease themselves. For example, a person might be tested for a gene associated with spinal muscular atrophy or Fragile X syndrome without experiencing any symptoms. The purpose isn't to diagnose them, but to inform them about their potential to pass the gene on. Beyond carrier screening, increasingly sophisticated genetic tests are available that analyze multiple genes simultaneously, looking for predispositions to complex diseases like heart disease, diabetes, or certain cancers. These tests, sometimes called panel testing, can identify individuals at increased risk. But it's crucial to understand that these tests often provide probabilities rather than definitive diagnoses. They highlight a higher likelihood of developing a disease, but lifestyle changes and ongoing monitoring often remain critical components of management. The role of genetic testing in prenatal care has also become a significant part of genetic counseling. Prenatal testing, including amniocentesis and chorionic villus sampling, can detect chromosomal abnormalities in a developing fetus. Counseling around these tests is particularly sensitive, as the results can be incredibly difficult to accept. Counselors assist expectant parents in understanding the implications of a positive diagnosis, such as Down syndrome, and explore their options, which might include continuing the pregnancy, preparing for the birth of a child with special needs, or choosing to terminate the pregnancy. Furthermore, genetic counseling plays a vital role in oncology, particularly in the context of tumor profiling. When cancer cells are analyzed for specific genetic mutations, this information can guide treatment decisions. Some cancer treatments are designed to target these specific mutations, dramatically improving outcomes. Genetic counselors help patients understand the meaning of their tumor's genetic profile and how it impacts their treatment plan. The interpretation of genetic test results is rarely straightforward. Many genes have multiple variants, some of which are benign, meaning they don't cause any problems, while others are disease-causing. The effect of a particular variant can also depend on other genetic and environmental factors. This is why genetic counselors emphasize that a genetic test result is just one piece of the puzzle. They work closely with physicians to integrate the genetic information into a comprehensive clinical picture. A critical aspect of genetic counseling involves helping individuals and families cope with the emotional challenges associated with genetic information. Learning about a genetic predisposition to a disease can be frightening, and it's natural to experience anxiety, fear, or uncertainty. Genetic counselors provide support and guidance, helping individuals develop strategies for managing their emotions and making informed decisions. They also connect families with support groups and resources. The field of genetic counseling is constantly evolving as new technologies and genetic tests emerge. It's a profession dedicated to empowering individuals and families with knowledge and support, navigating the complexities of our inherited genetic landscape with compassion and expertise. It's a reminder that understanding our past, our genetic blueprint, can help us shape a healthier and more informed future. The world of medicine has always relied on observation, experience, and a careful understanding of how the human body responds to treatments. For centuries, doctors adjusted dosages and therapies based on a patient's age, weight, overall health, and sometimes simply how they felt. But what if there was a hidden key, locked within our very DNA, that could dramatically improve the effectiveness of medications and dramatically reduce the risk of harmful side effects? This is the fascinating field of pharmacogenomics, essentially using our genetic blueprint to tailor drug therapies to the individual. It all begins with a realization we're not all created equal when it comes to processing medications. While a dose of a particular drug might work brilliantly for one person, it could be completely ineffective or even dangerous for another. This variation stems from differences in our genes, the instructions that tell our bodies how to build and operate. These differences aren't always major. Sometimes they're subtle shifts in a single gene, but collectively, they can have a profound impact on how our bodies metabolize drugs. Metabolism, in the context of pharmacology, refers to the complex processes our bodies use to break down and eliminate chemicals, including medications. Enzymes are the workhorses of this process. They're proteins that speed up chemical reactions, and many of the enzymes involved in drug metabolism are themselves controlled by genes. Now, consider this: some people have versions of these genes that produce enzymes that are incredibly efficient at breaking down a drug. For them, a standard dose might be plenty, and the drug will quickly clear from their system. Conversely, others might have versions of the same gene that create enzymes that are sluggish. These individuals require a much higher dose to achieve the same therapeutic effect. The most well-studied example of this phenomenon is the cytochrome PP450 enzyme system, CYP450 for short. These enzymes are involved in the breakdown of a huge range of drugs, from antidepressants to pain relievers to cholesterol-lowering medications. Variations in several CYP450 genes, like CYP22C19 and CYP2C9, directly influence how quickly a person metabolizes drugs like clopidogrel, cephalosporin, an antiplatelet drug, and codeine, a pain reliever. Some individuals carry a specific variant of CYP2C19 that results in a significantly reduced ability to activate clopidogrel into its active form. Without the active form, the drug won't effectively prevent blood clots. Conversely, people with a different variant of the gene might process clopidogrel so quickly that they're at a higher risk of experiencing side effects. The impact of these genetic variations extends far beyond simply adjusting a dose. It's not just about finding the right amount, it's about predicting whether a drug will even work at all. In some cases, a genetic variation can render a drug completely ineffective. Think of it like trying to start a car with a faulty ignition. The drug simply won't trigger the desired response in the body. Pharmacogenomics isn't limited to just CYP450 enzymes. Other genes play a role too. For example, variations in the VKORC1 gene influence a patient's sensitivity to warfarin, a powerful blood thinner. Warfarin's effects are highly individualized, and genetic testing can help doctors determine the initial dose needed to achieve the desired clotting effect, minimizing the risk of bleeding complications. Similarly, genes involved in the transport of drugs across cell membranes, like ABCB1, can affect how much of a drug reaches its target within the body. The process of incorporating pharmacogenomics into clinical practice is a carefully orchestrated one. It typically begins with a genetic test, often a simple blood test, to identify specific variations in relevant genes. The results are then analyzed in conjunction with the patient's clinical information, their age, weight, other medications they're taking, and their symptoms to predict their response to a particular drug. This isn't about replacing traditional medical judgment. It's about adding another layer of information to the decision-making process. As technology advances, pharmacogenomics is becoming increasingly sophisticated. Next-generation sequencing allows doctors to analyze a much broader range of genes simultaneously, providing a more comprehensive picture of a patient's genetic makeup. Furthermore, researchers are working to develop polygenic risk scores, which combine the effects of many different genes to predict a patient's overall response to a drug. The potential benefits of pharmacogenomics are enormous. It promises to reduce adverse drug reactions, improve treatment outcomes, and ultimately lead to more personalized and effective healthcare. It represents a shift from a one-size-fits-all approach to medicine to a system where treatments are tailored to the unique genetic fingerprint of each individual. While challenges remain, such as the cost of genetic testing and the need for more research, the future of medicine is undoubtedly intertwined with the power of understanding our own DNA. The story of DNA isn't simply a tale of blueprints. It's a saga of change, a chronicle of how a remarkably simple set of instructions blossomed into the complexity of life itself. For a long time, scientists assumed the genetic code, the relationship between DNA sequences, and the proteins they direct the creation of, was a fixed, ancient legacy, like a master craftsman's original design. However, as our understanding grew, particularly through the meticulous work in comparative genomics, a truly astonishing picture began to emerge. The genetic code has undergone a significant, and still partially mysterious, evolution. The initial wave of evidence came from studying the astonishing diversity of life on Earth. The sheer number of different amino acids, the building blocks of proteins, that organisms utilize was a critical clue. There are only 20 standard amino acids found in proteins across almost all life forms. Yet, different organisms utilize different codons, three-letter sequences of DNA bases, to specify these same amino acids. This redundancy, the fact that a single amino acid can often be coded for by multiple different codons, is a fundamental aspect of the genetic code's flexibility. For example, the codon UC almost always codes for leucine, but UG can also code for leucine. Looking at the simplest forms of life, the archaea, and the earliest bacteria offered further insight. These organisms, representing some of the earliest branches of the tree of life, possess a genetic code that is remarkably closer to the standard code we see in eukaryotes, plants, animals, and fungi, and even more complex organisms like humans. This suggested that the standard genetic code hadn't always been the case. It had, at some point in the distant past, undergone a significant shift. One of the most compelling lines of evidence comes from the study of alternative codons. Certain codons, particularly those that code for amino acids that are rarely used, sometimes appear to be encoded by different codons in closely related organisms. These variations, known as codon degeneracy, are not random. They are usually found between closely related species, suggesting they arose during evolutionary transitions. Scientists have identified several instances where a particular codon consistently changes in different lineages, reflecting a gradual shift in the genetic code. Several theories have been proposed to explain how this evolutionary change occurred. The RNA world hypothesis offers one explanation. In the earliest stages of life, before the widespread use of DNA, RNA may have served as both a carrier of genetic information and a catalyst for chemical reactions. RNA's simpler structure, compared to DNA, could have allowed for greater flexibility in the genetic code. Perhaps the initial genetic code was less rigid and gradually became more constrained as DNA evolved as the primary information carrier. Another theory, the translational frame shift hypothesis, proposes that a minor error in the way ribosomes, the molecular machines that translate mRNA into proteins, initially led to changes in the genetic code. Ribosomes don't read mRNA in a continuous, sequential manner. Instead, they scan the mRNA in groups of 3 nucleotides, as if searching for a specific pattern. A slight misreading during this scanning process might have introduced errors, leading to different codon assignments over time. This process would be amplified through successive rounds of protein synthesis, gradually shaping the genetic code. The proto-code theory suggests that the genetic code may have started with a different set of codons, perhaps one that utilized a different range of amino acids, or perhaps one that was less sensitive to variations in temperature or other environmental factors. This proto-code might have been more tolerant of errors, allowing for more rapid adaptation to changing conditions. Over vast stretches of evolutionary time, the codons would have been refined and stabilized, eventually leading to the code we use today. Interestingly, some researchers have discovered codons that are used in a non-standard way in certain organisms, suggesting that the genetic code is not as fixed as we once believed. For instance, some bacteria use codons to specify modified amino acids, or to stop protein synthesis entirely. These nonsense codons demonstrate the ongoing plasticity of the genetic code. The evolution of the genetic code isn't a single event, but rather a series of gradual shifts and refinements. It's a testament to the adaptability of life, a demonstration of how even the most fundamental aspects of biology can change over immense periods. Scientists continue to investigate these changes, hoping to unravel the precise mechanisms and timings of this remarkable evolutionary journey, and to further understand how this seemingly simple code underpins the breathtaking diversity of life on our planet. The story of the genetic code is, in essence, a story of life's ongoing experiment, a process of continual adaptation and innovation, written in the language of DNA. Deep within the cell, a quiet but incredibly powerful system exists, a mechanism cells use to carefully regulate their own activity, almost like a tiny, built-in editor. It's called RNA interference, or RNA, and it's a remarkably elegant solution to the problem of controlling which genes are turned on or off. For many years, scientists believed gene expression was primarily dictated by the careful orchestration of transcription and translation, processes involving DNA and proteins. But RI revealed a different layer of control, a way for cells to directly target and silence specific genes with impressive precision. The story of RNA begins with small RNA molecules, much smaller than the DNA that carries our genetic instructions. These aren't involved in building proteins, they're focused on a very different task, preventing the cell from ever reading certain genes. The key players are short strands of RNA called microRNAs, miRNAs, and small interfering RNAs, siRNAs. Both of these molecules are produced from longer RNA molecules called precursors. These precursors are transcribed from DNA, just like messenger RNAs, mRNA. But instead of being translated into proteins, they're processed into these smaller, active forms. miRNAs are often produced in response to environmental signals or changes within the cell. They're particularly adept at silencing genes involved in developmental processes, allowing cells to fine-tune their behavior in response to external cues. siRNAs, on the other hand, usually originate from outside the cell, often from viruses. When a virus tries to invade a cell, the cell can recognize its genetic material and produce siRNAs to specifically target and destroy the viral RNA, preventing the virus from replicating and causing harm. Once a mRNA or siRNA is formed, it begins a fascinating journey. These small RNAs patrol the cell's environment, searching for messenger RNA molecules that share a similar sequence. Think of it like a tiny detective, searching for a matching fingerprint. If a mRNA or siRNA finds a matching mRNA, it binds to that mRNA. This binding is the crucial step. It's like a lock and key. But instead of a physical key, it's a perfectly complementary RNA sequence. When a mRNA or siRNA binds to an mRNA molecule, it doesn't destroy the mRNA entirely. Instead, it marks it for degradation. The mRNA is broken down and cannot be translated into a protein. More dramatically, the binding of the small RNA can directly block the translation process itself. It physically prevents the ribosome, the cellular machinery responsible for protein synthesis, from attaching to the mRNA and building a protein. So, effectively, the gene is silenced, its instructions ignored. The beauty of RNA lies in its specificity. The sequence of the mRNA or siRNA determines which mRNA it will target. Because these small RNAs are perfectly designed to match specific sequences, they can silence only the genes they are intended to silence. This incredibly precise control is essential for maintaining cellular health and preventing errors in gene expression. Scientists have discovered that many genes are regulated by multiple mRNAs, creating a complex network of control. Some genes might be silenced by several different mRNAs, while others might be regulated by a single mRNA. This intricate web of regulation allows cells to respond to a wide range of stimuli and maintain a delicate balance between different cellular processes. Beyond understanding fundamental cellular mechanisms, RNA has become a powerful tool for researchers. Scientists can use RNA to study gene function by selectively silencing genes and observing the effects on the cell. It's like turning off one light bulb in a circuit to see how it affects the others. This approach has revealed insights into a vast array of biological processes, from cancer development to neurological disorders. Perhaps most remarkably, RNA is being explored as a therapeutic strategy. Researchers are developing methods to deliver siRNAs or mRNAs to specific cells or tissues to treat diseases. For example, in cancer research, scientists are using RNA to silence genes that promote tumor growth or to make cancer cells more vulnerable to chemotherapy. Similarly, in infectious diseases, RNA is being investigated as a way to combat viral infections by directly targeting viral genes. The journey of RNA is still unfolding, and scientists are continuously learning more about this remarkable cellular mechanism. It's a testament to the incredible complexity and ingenuity of life at the molecular level. The ability of cells to so precisely control their own genetic destiny highlights the ongoing dialogue between DNA, RNA, and protein, a conversation that dictates the health and function of every living thing. It's a quiet revolution happening within us all, a constant reminder that even the smallest parts of our cells hold the keys to understanding the greatest mysteries of life. The earth holds secrets, silent and deep, and sometimes those secrets whisper through strands of our own history. We've learned how to listen to them, not with our ears, but with incredibly sensitive machines that can unravel the story held within the ancient DNA of fossilized remains. It's a remarkable process, a delicate dance of chemistry and technology that's allowing us to connect with extinct creatures and, in a profound way, with our own ancestors. The key to this endeavor lies in the surprisingly resilient nature of DNA, even after thousands, sometimes millions of years. DNA itself is a fragile molecule, a long, twisted ladder built from just 4 chemical bases: adenine, thymine, cytosine, and guanine. These bases pair up in a strict, predictable way: adenine with thymine and cytosine with guanine, forming the rungs of the ladder. While this structure is remarkably stable, it's still vulnerable to damage from radiation, heat, and enzymes. The longer DNA has been buried, the more degraded it becomes, transforming from a recognizable ladder into a tangled, broken mess. Early attempts to extract DNA from fossils, dating back decades, were largely unsuccessful. Scientists used techniques that were simply too crude, too aggressive, to avoid completely destroying the precious genetic material. However, over time, advancements in molecular biology and analytical instrumentation have revolutionized the field, leading to increasingly successful ancient DNAs, a DNA analysis. A pivotal breakthrough came with the development of new extraction methods. Instead of relying on harsh chemicals, researchers now employ milder, more targeted approaches. Initially, a freeze-dried powder of bone or tooth was created, removing water to stabilize the sample. This powder is then carefully treated with enzymes, proteins that catalyze biochemical reactions to selectively break down the surrounding matrix of the fossil, like the mineralized bone, without damaging the DNA itself. These enzymes are carefully chosen and controlled to minimize collateral damage. The next stage involves a process called DNA amplification, using a technique called polymerase chain reaction, or PCR. PCR is like making countless copies of a specific piece of DNA. A small, degraded fragment of DNA is used as a template, and enzymes, along with short DNA sequences called primers, are used to build new strands of DNA that are complementary to the original. This process creates exponentially more DNA, allowing scientists to work with a much smaller, more manageable sample. Once amplified, the DNA is analyzed using next-generation sequencing, an NGS technology. NGS allows researchers to determine the exact order of the bases within a DNA fragment. It's a process akin to reading the entire sequence of a very long book, letter by letter, and identifying each word, base within it. This process is incredibly detailed, allowing scientists to identify mutations, differences in the DNA sequence that have accumulated over time. The data generated by NGS is then compared to the DNA sequences of modern organisms to infer evolutionary relationships. For example, a DNA sample recovered from a woolly mammoth tooth could be compared to the DNA of modern elephants and mammoths to determine the evolutionary lineage and estimate the time since the mammoth went extinct. The more similarities a DNA sequence shares with a modern relative, the closer the evolutionary relationship. However, it's important to note that aDNA is often highly fragmented, meaning that only small pieces of DNA are recovered. This makes interpretation more challenging and can lead to uncertainties. The study of aDNA has provided astonishing insights into the past. For instance, scientists have been able to determine that Neanderthals and Denisovans are extinct human relatives, interbred with early modern humans, leaving traces of their DNA in the genomes of people alive today. The presence of Denisovan DNA, for example, has been confirmed in the genomes of individuals from Southeast Asia, impacting their immune system and adaptation to local environments. Furthermore, aDNA analysis is being used to study extinct animals like the saber-toothed tiger and the giant ground sloth. By sequencing their DNA, researchers are reconstructing their evolutionary history, understanding their diets, and gaining a deeper understanding of the ecosystems they inhabited. The preservation of aDNA in permafrost regions, like those in Siberia, is creating unprecedented opportunities to study ancient life forms that were previously inaccessible. However, working with aDNA presents significant challenges. Contamination is a major concern. Even tiny amounts of DNA from the researcher's own body or from the environment can introduce errors into the analysis. Strict protocols are followed to minimize contamination, including working in sterile environments, using dedicated equipment, and employing decontamination techniques. Another hurdle is the age of the samples. As DNA degrades over time, it becomes increasingly difficult to distinguish between genuine ancient DNA and contamination. Despite these challenges, the field of aDNA analysis is constantly evolving, with new technologies and methods being developed to improve the accuracy and reliability of the results. It's a testament to human ingenuity, a way to listen to the silent echoes of the past, and to appreciate the interconnectedness of life on Earth. As we continue to unlock the secrets held within ancient DNA, we gain not just knowledge of extinct species, but also a deeper understanding of our own place in the grand narrative of life. If these explorations offer a quiet moment, please subscribe and share them with those seeking serenity. We've journeyed through the intricate world of DNA and genetics, unveiling the secrets within. Allow your mind to settle, your thoughts to flow gently, without urgency or concern, until sleep embraces you.